Disc 12.
The 1950s saw the development of cytogenetics, the study of chromosomes through the microscope. Employed diagnostically, this approach soon revealed that abnormalities in chromosome number, usually one too many or one too few, invariably cause profound dysfunction. These problems stem from an imbalance in the number of genes, a departure from the norm of 2 of each. Such conditions do not run in families, like Duchenne muscular dystrophy, DMD, or cystic fibrosis, CF. But they are still very much genetic. They arise spontaneously through accidents in the cell divisions, leading to the generation of sperm and egg cells. The best known is Down syndrome, named for John Langdon Down, who in 1866, as the medical superintendent of a home for the retarded, was first to describe its characteristic clinical features. He noted that 10% of the residents of his institution resembled one another. So marked is this that when placed side to side, it is difficult to believe that the specimens compared are not children of the same parents. But the first insight into the condition's biological basis did not come until 90 years later, when the French physician Jérôme Lejeune found that children with Down syndrome have 3 copies of one chromosome, later shown to be chromosome 21. The normal condition, 2 copies of a chromosome, is called disomy. So Down syndrome is known in genetic parlance as trisomy 21. The incidence of Down syndrome increases with the age of the mother. At age 20, a woman's chance of producing a Down baby is about 1 in 1,700. But at 35, it jumps to 1 in 400. And at 45, shoots to 1 in just 30. For this reason, many older pregnant women choose to have prenatal diagnosis performed on their fetus to determine whether it might possess 21 in triplicate. The test was first done in 1968, and today it is routinely offered to all pregnant women over 35. Because the developing fetus must be big enough to withstand safely the extraction of a tissue sample, such diagnosis cannot be performed in the very early stages of pregnancy. Typically, it is done in the 15th to 18th week using amniocentesis, a procedure that entails drawing some amniotic fluid, which naturally contains cells from the fetus. An alternative test, which may be done as early as the 10th week, gathers cells from the chorionic villus, the part of the placenta that attaches to the uterine wall. But this method is less reliable. Because both procedures are mildly risky, amniocentesis results in a 1% rate of miscarriage, and chorionic villus sampling in a 2% rate, younger women are generally advised to avoid them. The probability that their fetus has a genetic defect is actually lower than the probability that it will be damaged by the procedure. At one time, extracted fetal cells had to be grown in petri dishes before being processed for chromosome analysis. Nowadays, a more rapid diagnosis can be done using fluorescence in situ hybridization, FISH, or FISH. In this method, a small fluorescent molecule is attached to a stretch of DNA sequence specific to chromosome 21 and introduced to the sample, where it binds to the fetal chromosome 21 DNA. If 2 fluorescent patches appear in the nucleus of a cell, the fetus is normal. If 3, the fetus has Down syndrome. In Britain, 30% of Down pregnancies are detected by routinely testing the oldest 5% of women bearing children. This method boasts a clear efficiency in simple terms of detections per pound spent. Britain's National Health Service has been subject to such a calculus ever since Mrs. Thatcher's assault on health spending. But what of the remaining 70% of Down cases? Down is rarer in the babies of younger mothers, but these women account for the vast majority of all pregnancies. Since the standard tests are statistically not worth their attendant risks, there have been attempts to find alternative, non-invasive indicators. It turns out that substances detectable in the mother's blood yield useful information. Low levels of alpha-fetoprotein and high levels of chorionic gonadotropin correlate to a significant degree with Down, though they are by no means ironclad indicators of trisomy. Modern practice, then, is to offer younger women the blood test, and if it suggests the possibility of Down, they are then counseled to undergo amniocentesis, or chorionic villus sampling for a definitive diagnosis. Sadly, today a woman who learns that her fetus has Down syndrome has only two choices: to become the mother of a Down baby or to abort the fetus. It is a painful decision that is made no easier by the variable severity of the affliction. People with Down all share the characteristic facial features identified by Dr. Down: a broad, flat face, a small nose, and narrow, slanting eyelids. But they range considerably in IQ, scoring between 20 and 85, i.e., from severely handicapped to low normal. Dr. Down originally called the disorder Mongolism on the basis of these characteristics, entitling his 1866 paper Observations of an Ethnic Classification of Idiots. Subscribing to the racist evolutionary views of his day, he believed that Down represented an evolutionary step backward from the exalted Caucasian state to the inferior Mongoloid one. To give him his due, though, he concluded that what he called retrogression undermined the claims of those who refused to accept that Caucasians and non-Caucasians were members of the same species. Down syndrome babies are especially prone to a range of ailments, including heart disease, which claims about 15% in the first year of life, gastrointestinal anomalies, leukemia, and with increasing age, cataracts and Alzheimer. But it's also perfectly possible that an individual will have relatively few health problems. With improved care and better knowledge of medical hazards posed by possession of that extra chromosome, life expectancy has increased substantially. 50% of affected individuals today survive into their 50s. Despite typically acquiring over time what most would consider a depressing familiarity with the insides of hospitals, people with Down generally enjoy life and have brightened many a family. The condition is perhaps tougher on their parents, who must adjust to caring for someone with special medical needs, as well as to the knowledge that their child will, in many ways,